how how the sequence are getting aligned, how the sequence are getting trimmed, getting filtered, and then it is how getting aligned with the reference genome. Once the sequence is getting aligned properly with the reference genome, we need to call the variants. The main important steps of the NGS data analysis is calling the variant, and then once the variant is called, we need to annotate that. That is what we are going to see today and tomorrow with the continuity of what we are going to see. So, what is the main importance of calling the variants and annotating them? So, variant calling and variant annotation are the very, very crucial process in genomics. As told, what is the main theme? It is helpful to identify the genetic markers in a particular individual, and then that is moving into the interpretation, how that is making the changes with the real-time steps. So this the main importance of identifying the variants, calling the variants, is to understand how the genetic diversities are, how it is diversely changing from uh, one position to another, how that is causing the diseases. Once that is caused the disease, to diagnose what type of diseases and what is the advancing personalized medicine for the core complex diseases we can do, and how can we drive in the scientific discoveries, new discoveries happening the real time. It's to know all this, the variant calling and variant annotation is very important. See, this is just an example. It is from an IGB viewer where you can see the set of genes positions and genes, how the variations happens at each uh, organ, sorry, each species, each samples. So, how what are the different mutations happening? The red color, green color highlighted the changes, what it has happened. To know, to understand what is meant by the main importance of to understand the genetic Indels, structural variations and copy number variations all together happen. That is helpful to study a population genomics, and then to do a functional studies where we can link specific variation to a specific gene regulations and protein functions. So here is an example, different types of variants. So if it is take, it is the reference read. Reference reads have the variants at A, but when we are seeing the following sample reads at the particular position, instead of A, it is changed into C. But it is in the reverse stand, it is not in the forward stand. So it tells like this is non-variant. What we mean by SNP? SNP is called a single nucleotide polymorphism, the change of only one nucleotide. Similarly, in the reference position, instead of the reads, the sample reads is changed into C, but you can see it is changed the single nucleotide, it changed in multiple samples. So that is telling like single nucleotide polymorphism. That is a SNP. That is the first and foremost genetic variation. And the second most thing, insertion. So here in the reference, T is there, but in the particular position, instead of T, the I is getting inserted. The new stuff is getting inserted. So I, instead of T, that is new thing is getting inserted. So that is called insertion. Deletion means there are in the particular reference position, ATCG should be there. AGT will be there, but in the samples it is only A. GT got deleted. So that is called deletion. This is how the genetic variations are look like. So in a see in a condition like genetic identifying the genetic muting in a disease researchers, it is disorders, genetic disorders, this is called seen the genes are genetic disorder or that kind of a very complex and diseases where the NGS variant calling is helpful. And foremost thing to study cancer genomic related disease where it is detecting somatic mutation driving tumor processions as KRs and EGRF mutation to study that tumor suppression gene list where it is helpful in that disease research. In the field of precision medicine, as you all know from the beginning, the NGS is mainly focusing on, it is one of the main application is to do a precision medicine study. The tailored treatment is based upon individual genetic makeup, where if you take pharmacogenomic study, it determines drug efficiency and safety to that which is helpful to that is affecting uh, clopidol metals that are linking variants to the phenotype. So if the variant is with this mutations, a specific variant changes how that particular way how the lot of Mendelian inheritance pattern and polygenic risk scoring steps. Moreover, that it is helpful to prioritizing the variant. When we focusing on a clinical or a functional significance of a particular variant, that variant, some of specific variants or the specific gene, which is filtered and came into a prioritization, that is helpful to study the validation of a therapeutic targets. Like if you're working with the oncology-based study, if some gene like BRCA1 or BRCA2, you also know, like have breast cancer genes, which will which is getting priority, most priorities. That is helpful to reduce if the tumor is getting suppressed, the specific gene is getting suppressed. We need to give a proper functionality to a particular set of genes to study targeting therapy, what kind of study we are working with. Not only that, it is also helpful to reproduce the research in standardizing the data interpretations across the studies. The first and foremost thing is facilitating comparison between data sets and ensures the consistent variant annotation. So using we need to do the annotation. So using tools like snippet, ANOVA, or ensemble VEP variant effectors predictor, where we can able to reproduce the annotated results and how it is clinically or socially getting impacted. For example, if you are working with advancing the genomic medicine, the first and two or three is clinical diagnosis and therapeutic development. So that is helpful in a clinical way for all the physicians or the clinicians who is working to improve the diagnosis of genetic disease, and with that is also helpful to guide a specific procedure based drug design. It is helpful for improving the agricultural practice, and it is also helpful to support public health initiatives. So this is now we are going to see about the different types of variants. So as told early to the multiple times, but to know the single nucleotides suffix, so I'll give you very few. Single nucleotide polymorphism is a variations in us of a single nucleotide at a specific position. It's just one nucleotide is changing at one position. So here in the reference, AAGCT, instead of A, the C is replaced in the one one sample. So that is called variation. 300 is getting happening. So you should understand in the human sample, for every 300 bases, one single nucleotide, that is it's okay, one mutation is getting happening. So this is another image similar to how insertion deletion taken place. And the types of variation. One is synonymous variation. Synonymous variation means the amino acids of the particular variation does not change. It remains similar, which is SNPs or regulatory that is that is affected after the gene expressions. So the significance of this particularly, like, yeah, ApoE SNP that is linked to a Alzheimer's disease. This is just so another type of mutation is called indels. Indels is called insertion. What's the issue? Who writes that? What's the issue? Okay, nothing. Okay. So the insertion and deletion of a single nucleotide sequence in the DNA sample. So here is the reference genome where ACTG is the reference genome, and you can see insertion that is the next to CG is getting added, that is ACGTC, that is called insertion, and you can see C is getting deleted from the reference genomes, but not in the reference genome in the sample of what we are working. So that is called deletion, where one particular nucleotide is getting deleted. So that can be focused usually a few bases per, but can be extended to a large bases. It can be in a few bases only, like 300 within 300, but sometimes it can be a large base pairs also getting deleted. The insertion and deletion can be of two types. One is frame shift indels, another one is non-frame shift indels. What is called frame shift indels means not in multiple. What is mean by multiples of three? Here you say ATCG, ACTG. Instead of C, one is that one C is getting deleted, right? In not it's not will be like after three bases once again get deleted, but what a non-frame shift indels means in a multiples of three, like after 3% the model progress steady. You can reference and the red one is a sample of our interest, that is very in genome. You can see which is highlighted in the white, the nucleotides are getting changed. So that is called SNPs. You can see in this position deletion has taken place. This is called small deletion, and this is position insertion has taken place. This is called small insertion. So you can see if in the reference at this particular question where the where this is got deleted, and in blue is the reference that new arrow mark was getting added, where that is called insertion. Tandem repeats is you can see in the reference only one set of pages are there, but the same one is getting duplicated two times in the uh, sample of our interest. So that is called tandem repeats. That is happening two times in the same set. Interspect duplication means with the space, with the last set of deletion, its particular position, the same set of genes getting duplicated in the mm, getting duplicated in the following data sets. Then inversion and transversion, transposition. Inversion is like in the reference genome, if it's the forward strand, that is called it will be the forward strand, but in the sample of our interest, it will be in the reverse strand. Transpositions, it will be in the two types. One is it is yeah, you can see here introspective duplication. In the reference genome, it must be in the one position, but if it in the sample of our interest, it will be in the different. Mm, as I told, it is happening. The segment is getting twin. Another thing is called translocation, where the segment of DNA move to a different chromosome. See, that is quite completely changing. If you take an example, chronic my, myelin leukemia, that is called the Philadelphia chromosome, which is results from a translocation between chromosome 9 and 8. That is completely changed. The segment is completely changed from chromosome 9 to chromosome 22. So here you can see short read signatures of structural variants. This is deletion, duplication, insertion, transversion, translocation. So here is what is this? It's in the our genome sample is in the light turquoise color, and then if you take inserted sequence, it is in the blue color. So the first one, deletion. In the reference genome, there is a three set of pairs. But in the sample genome, one is got deleted. So that is called deletion. So the forward strand is in the green is called the paired end. The red one is three strands is duplication. What's this? Hess Asiba, yeah, tell me what's your question. Sorry, who's this? Madziba. You have any questions? You can ping in the chat. I'll answer. No questions? Okay. What happened suddenly? I don't know. So this is how duplication has taken place. What does mean by insertion in a condition like in the reference genome that is new, that is nothing is there, only two set of pairs, or read one, read two, read three. But in a case of insertion, there is new center one getting added. That is called insertion. But in an inversion means it is in the reverse position. That is called inversion. Translocation, which I already told. So deletion duplication means a segment is getting copied, resulted in extra copy. That is duplication. Deletion means a large region of DNA is missing. That is deletion, and that significance is leads to genetic disorder or cancer or evolution. SVs can be interpreted, and that is helpful to lead the phenotypic changes. The copy number variations. The most important two type of variation. One is structural variant, another one is copy number variant. So copy number variant is it's a variation in the number of copy of a specific question that is or often involves either in a deletion or a duplication. A normal genome are two copies of genes, one from each parent. Uh, in a copy number means one from each parent, right? Three copies duplication or one copy are getting deleted. So the length is larger than 1 kb but smaller than chromosomal abnormalities. If it is a chromosomal abnormalities, will be little high, but it won't be that much having a chromosomal abnormality. So if you can see in the chromosome position, the control, the parent have A, B, C, D, but the deletion happening means B is getting deleted. Duplication means B is added twice. Inversion means it is in the different position. Insertion means it is getting added. So this is all about copy number variations, where the main thing is it influences multi-number variation is happening to cause the diseases. So it can be helpful to study a large amount of neurodevelopmental or psychiatric diseases. What is the nature of types of disease? How significant it is? We take Indels. What is insertion or additions? SVs are structural variants. SNVs is called the 1 kilo basis, copy number changes, and how that is significantly affecting the different areas. That is what you had it here. So how it is getting applicable? So as I told, it is helpful for personalized drug discovery. in a form of pharmacogenomics, cancer therapy, and rare disease treatment. If we take disease research, it is helpful for monogenic disorders, cancer, genomics, and complex diseases. It is also helpful in evolutionary and population-based genomic studies, where it is helpful for ancestry and migration, or helpful to reconstruct a population history, the evolutionary history, to study that the variant calling and the variant annotation is helpful. And also it is helpful to understand the genetic adaptation, where how to identify specific variants, how that variance is getting survival and in the specific environment. And also it is helpful in agricultural and environmental genomic study. Reproductive health, specifically to see pre-implantational genetic testing to study either identify any pathogenic variants present in the embryo, which is technical algorithm to perform and replicate sequencing to confirm the findings whether we have done properly or not. There may be a chance of low sequencing depth. It may be if the coverage is less than 30x. During that time, there may be there is a lot of chance like we miss the variants, specifically for a rare or a low frequency mutation in that particular kind of control. So if the depth is below 30x, if it is somatic mutation, if the depth is below 100x, we'll feel the issue. And mapping errors, while mapping with the reference genome, there is a lot of misaligns will take place. During that condition, we need to use proper reference genome, proper analyzing tools. And when we are doing a large set of data with large amount, we might miss storage or pre-processing might of stuff. That is also a technical issue we face. In a computational issue, challenges will be a complex workflow standard tools like GATK, Docker. Another thing like incomplete database, when we are doing annotation with stuff, the databases which is publicly available might not be getting up to date. During that condition, we must to identify the novel variants which is identified by us. And complex variants, if we are having a large amount of structural variants or copy number variants, during that time we might able to difficult to detect with the tools which is already developed. In that kind of condition, we might miss to identify a proper. The variants might causes the help might miss to identify the rare variants. So we need to use very proper targeted depth sequencing for a specific region. Population in all population, but we are doing with the population-based study. While doing lack of context, if there is variance may lack annotation details, that is oh, the website is so low, or my internet is low, or I don't know. Any questions? Then let me continue. Next, we are going to adding into the stuffs. When we are working with variant calling, we need to see three things. One is sequencing depth. Sequencing depth is also known as coverage, which is referred to the number of times a particular set of genomic position is sequenced during the sequencing process. It should be of two types. One is high depth and low depth, and it should be in a proper coverage. If it is germline variant, it should be above 30x. If it is somatic variant, it should be above 100x. Is all about sequencing depth. So to calculate the depth of human sample, here is the formula. Depth is equal to total base sequence divided by human, so genome size. So here, if the human genome size is 3 billion bases, that is can be helpful to produce 90 billions of data. So 90 billion by 3 billion is equal to 30x. That is the base set of data to do the analysis. Then quality scoring, where quality score represents the confidence in the accuracy of each base calls in the sequence trees, which is typically encoded in the Fred format. So if you take a Fred quality score, the formula Q is equal to minus log 10p, where p is the probability of bases is incorrect. If the Fred score is 20, there is 1% of chances of error. If it is 30, then will be a 0.1% of chance of error of 99.9% of accuracy. To encode in that, the quality score are stored in ASCII characters for giving space efficiency. If it is Fred plus 33 encoding, it means it is in the modern term of FASTQ files. It is in the range quality of value, which is above 30x. The main importance of doing variant calling, that of two types. One is low quality score, that increases the likelihoods of false positive or false negative. That can be checked properly with of databases called GADK and SAM tools, which is uses the quality scores to filter unreliable variants. Then what is called reference genomes. So reference genomes are a curated sequence of a species genome that is used as a baseline for comparing individual genomes, genomic sequences. For human reference genomes, there are two types. One is HG37 and HG38. HG37 is the older version that is also called the HG19, that is also called GRCS37, and GRCS38 is the latest version with a better resolution in the uh, complex region. The main importance of using variants, one is of two types. One is to do the alignment, another tuning is used to do variant calling. Challenges will face is two things. One is population biases. Like if you are doing a population-based study, it lacks the representation of diverse population. And if it is having gaps and errors, the regions like telomeres or centromeres may be incomplete or misassembled. The advantages is graph-based reference genomes is all about. Next, we are going into the entire workflow of how the variant calling has taken place. The first main thing is we have a reference genome, and then we have the NGS rates. Then if you have two things, we need to do the alignment. The alignment can be done with BWA or BOP. Once that alignment has done, we'll get the data in a SAM format. Once the SAM data can be converted into BAM format, and then we need to sort the BAM file. Once we hit is done, we need to mark the duplicates and need to do the realignment. Once realignment all done, we need to do a recalibration if the base quality score is less. Once all this yellow stuffs, remove the duplicates, marking everything has done, we need to see call the variance, and we need to do a variant recalibration. So the process pre-processing, as you all know how the pre-processing has taken place, we need to do a quality check, we need to do the trimming process, and we need to do the alignment stuff. And the post-alignment stuff also we have done. The now major stuff is to identify the genetic variants. The three major steps or tools is one is GATK haplotype caller, another one is free base, another one is SAM tool, BCF tools, and another thing is called deep variant that is AI based. So this all the different tools are helpful to helps to identify call the variants, and that will end product as generate the VCF file as the output. The raw VCF file as the end output. Once we'll get the raw variant file, we can able to identify some of the low confidence variants, and we are our aim is to focus on high quality results. In that particular kind of conditions, we will apply quality threshold metrics like depth, quality by depth, and strand biases. That can be done through a tool called GATK variant filtrations or VCF tools. Then that is helpful to filter out false positives using strict criteria. And at the end, we'll get the filtered VCF file, which is contains the only high quality variants. The low quality confident variants are getting removed. And once the variants are getting filtered, we need to annotate the variant. That is adding a functional or clinical context to identify the variants. That is of three types. One is ANOVA, another one is SNPF, another one is called Ensemble BEP. uh, but that can be identified and checked with the external databases like ClinVar, DBSNPs, Cosmix to predict the clinical variants. And as that, we'll get the annotated VCF file as the end output. So this is all about the complete final step of whole genome sequencing. That non whole genome sequencing, that is next generation sequencing. Here is the Pakka work table where you can explain how you can do, what are the different stages, what are the different tools, what are the different output you can get. I'll explain the pipeline how to work and all. So far theoretically you can able to see how this is happening everything. This is the best table you can have it as a picture in your phone when we are working, when you are understand. Oh, this is the very basic to advanced strongest flow tools which is working worldwide, because I have experience of work with all the stuffs. So I prefer to take you to understand this is what you are working. Any questions so far, or I'll go to the workflow stuff, how the different tools work. No questions, all clear, all fine with it. Shall we go and see what are the different websites, how it works? Shall I continue? Shall I start? Is anyone is there or all went to sleep because had college? I'm back and listening to the course. Now we are going to see about different tools with the different data sets. With the continuity of what you worked yesterday, you can continue with the variant calling process. So for sure today I'll share yesterday PPT, but tomorrow I'll share yesterday today and tomorrow PPT. Okay, no response, then I'll feel like I'll start continue. So to call the variants, there are four important tools we'll work with. The first and foremost tool where will preferably work worldwide is GATK. Another thing is free base, SAM tools and deep variant. GATK is called Genome Analysis Toolkit, which is very comprehensive toolkit that is having the inbuilt command pipelines variants. It supports both germline and somatic calling. Used that is to identify the variants is called haplotype caller. It is a where it is base quality score quality score recalibration. It is also helpful to do joint phenotyping across multiple samples. If we have thousand set of samples, when we want to call the variants with similar to all the stuff, that kind of study where GATK is the strength. Then in other conditions like it is having a highly accurate SNPs and Indels, it is designed for larger scale data sets and or do. So let me take you to the website called GATK. So my God, I'm extremely sorry. Genome Analysis Toolkit. If you go into the website, you can see the user guide, technical start, documentation, everything from in and out, how to download the latest version. Here, if you go with the user guide, with the documentation stuff, you can able to see everything from basis to do what is the how to do trouble stuffs, how are the different calories, how we can do interpret the SV structural variants of ECF files, and what are the different type of tests it is having inbuiltly having. We to go into tool inbox, so index you can see how the tools work. That is the flow. I have missed it. Get started. You can see you can go how to get started with GATK. How about the best practice method? What is the difference between germline, single variant, or double variant? So how to the analysis phase, pre-processing. What is the experimental design? How the scripts. Everything you will see in step by step. In the guide also you can go and see calling the variants, everything you will be added already in the PPT. Sorry, in the website itself. It is a best example. You can see how the workflow is, how what are the steps, how the workspace, how does the pipeline. In a case of the sample data, you can have the sample set of data we can work with, and what are the input lines, output lines, how everything it works, you can able to see. It is just a software tool, but it has got implemented with Galaxy that you can do from your end. So once you call the way, once you did the alignment, you can able to do calling the what calling the variant, just the tool which is implemented with Galaxy, integrated with Galaxy. You can use that for the analysis. So to run GATK, you just need to download GATK, and you just need to the give the command to call the haplotype for a single sample. This GATK, which is downloaded, and then is haplotype caller. In that, we need to give hyphen har reference pasta. Input is the BAM file, which is getting sorted, which the where it is get recalibrated, where we'll mark the duplicates. After all that, we'll have that one BAM file, right? That that BAM file we need to give it as input. As the end, we'll get it the output in the PCF format. It is helpful to do for a single count. A set of sample or write a different set of two. In doing a parallelization, you can add the command instead of one input. You can give the location of last from each set of data, and that will end in the results of getting results for each set of sample. What does mean by joint genotyping? That we are joint genotyping is generally to identify the genetic variants which is common in multiple set of sample. To do that, the GATK by default having their own uh, protocol or a program or a command or Python, sorry, command or a script. Just like GATK, genotype, GVCFs, reference faster we need to give if you are working with human genome. If you, only human genome, they will do joint genotyping. So human reference genome and sample one, that is V1, V2. So if you have multiple set of sample, we need to give you V1 to VN series, and that is helpful to do joint outputs. This is how the GADK works. If you want to work with free bases, free bases is developed. That is also helpful to identify the haplotype. That is also for both germline and somatic variants. The main thing of the free bases is also identify the haplotypes. That is how going to identify the relationship between SNPs and Indels. It does support for all the studies, multiple samples, population-based analysis, we can work with both diploid and polyploid based genomes. So the limitation is then GATK, because GATK is the most standard uh, variant calling pipeline, which is using over complex region. To run similarly, we need to download free bases. Once we then download the free bases, we need to give the reference faster, input file and the output file. This is generally single command, right? But we are running, it will take long time. We cannot run it in the laptop or any kind of small machines. We need to have a proper HPCs, like platforms. You need to have a slumber like that, or if you have a proper Linux with the high storage, you can work for a small set of sample. You can't work in human samples in Linux systems and all. What does mean by SAM tools and VCF tools? SAM tools and VCF tools are also having a variant calling and variant manipulation tools. We can combine the read depth and quality metrics for SNPs and interlinks. So it is integrated with the BCF tools, file manipulations like sorting. So the limitation is not as sensitive, as strong as GATK or FreeWise for complex data. So I don't prefer samples of BCF tools for calling the variant, but sometimes when doing other sorting, indexing kind of stuff, it is very helpful. So here I also helpful if some people is not getting able to download um, what is that, SAM tools or BCF, so BCFs too, sorry, GADK or free basis, they can work with that. So it is helpful that BCF tools yum pile up. You need to give it this just BCF tools m file up reference faster input, and we get it and the output file. If you want to call the variants, we'll use BCF tools call -mv or is it output faster and the input is raw PDF file. Another, the most likelihood of uh, working with uh, stuff is called deep variants, where it's a machine learning based variant calling, which is developed by Google specifically for a highly accurate SNPs and Indel detection. The main aim is to utilize the deep learning process to interpret sequencing data. It supports whole genome, whole exome, and whole targeted sequencing data sets from any platform. provide very accurate data, but it's now it's widely using. But I don't have had experience right now. But I need to work. Very soon, I'm going to work with deep variants soon. So I'll get better idea. So this is request GPU acceleration optional for optimal performance. That is also we need to download. So we need to download in a command line platform, and that is deep variant. You know right how to download the get command. So you need to download. Once that is downloaded, you can do run a command like run deep variant model type. What type of model we are, so sequencing model we have worked here. They have mentioned WGS reference. We need to give the reference. We need to give it as. Then here num shared is like how many number of node you are getting going to be used, or the number of shared is there. To add that, if we here they have added num shared. So this is the best way to use which tool for high accurate and standard workflow for larger project. Yes, Monica, you have any question? You can ping in the chat. I'll answer, or you just join the class. Any question, Monica? No. Okay. I don't know what happened to you all today. No response. Okay. Then once we'll mark the duplicates, we need to do our recalibration of the base quality scores. The base score is have is already having. Once we have the base quality, we need to do our recalibration. So that can be done with GATK, but we use GATK based recalibrator with the reference in input, and the reference FASTA file we need to give, and the input file will be the uh, deduplicated reads in a BAM format, and the known sites that is from DB snubs we need to give it. We need to already give, and also we need to give the recalibration table data, that what table to know, to let the machine knows how it need to be processed. So we need to all this, but this is just a base command. But when we are working with a different set of data sets, it will be getting deferred. Once this all done, we need to call the variants. That can be done with a command called haplotype caller. So the haplotype based calling methods that is helpful to identify the haplotypes that reads, and that can be in a format of GVCF format for doing a join calling. So here is a command GATK haplotype caller reference FASTA input is recalls read spam, output is VCF.gz and ERC GVCF. So end output it should be GVCF. Once we'll get the GVCF, we'll do the join calling, join phenotyping. The join phenotyping with list of so here you can see here I have given the text file that should be in a particular folder. That list location has been given, and then we call the joint genotype variant that will come with the end output into the single VCF file that have the variants from all the samples from the all that. Once we'll do the joint genotyping, we call joint genotyping is only for human sample. Please understand, it is not for all the variant samples. If your objective requires joint genotyping, you can work based on that. In the end, we are going to see the variant filtration where we'll helpful to reduce the false positive and improve the accuracy. In that condition, based on quality based thresholds, will do a variant filtration that can be done with GATK variant filtration. R is reference FASTA, V is rar RAR variant we have downloaded, and the filtration done with the quality depth less than 2, F is above 60, and the filter name is low quality filters. We need to remove all this. So for that condition, we are giving the filtering requirement over here, and as the output you can able to see filtered variant VCF. This is all about filtration. So here is a command with an example you can run once you have the data set. If you are running with a single data set, you can run it like this. So R is capital R is called path to the reference genome. N is the path to the input BAM file, and capital O is called output reference genome. Sorry, output VCF file. GVCF, if you want to get GVCF, you need to give ERC GVCF. ERC is called enable the GVCF mode. That is called ERC. Output is g.vcf.g is a genomic VCF file containing genotype likely hertz, haplotype with additional parameters, scenarios. First one is specific to a region of interest. We want to see specific regions. Through we need to give the specific regions intervals, chromosome position in detail. That can be given in an interval.bed file where it have a chromosome and a gene list, which is using a predefined panel of knowing variants. Like if we have a specific set of gene list, we want to know that gene specifically from all the set of sample. In that condition, we need to give it dbSNPs VCF file, and we'll get the sample variants. So dbSNPs is letting reference VCF with the known variants. If we want to do a parallelization for a large data set, that means what is mean by parallelization is the genome gets splitting into small chunks that scatter across different techniques and do the process individually. So the first main step is we need to split the intervals. That is reference genome. L is we need to give all the bed file, whole genome bed file. We need to call the variants in that time. And so this is not much needed in all. So, what you are thinking right now? How it's going into mind? Will it be very hard, or do you feeling like it will be too fast to learn? Something kind of that. What does anyone let me know? Any reply? No response. I think all asleep. Okay, no response. So, so other tool is called BCF tool on free basis. It is also command line based tools. So BCF tools is to mainly to manipulate BCF files and BCF file to perform variant calling, filtration, and annotation. So here I have given the commands and the process how to do filtration with the data sets of what you are having already. So BCF file, M pile up, OU reference faster, input BAM, VCF call. We need to call the VCF. So that can be map output uncompressed VCF format for piping. FNF is called reference faster. I is input PAM. VCF calling as MU. Or is it MU is like MV is calling variants in a multiple alignment mode, or is that it's a compression output data zip format format, or is that is generally output in a zip format. And we can do filtration based upon quality score is less than 20. We need to remove all that, and depth is less than 10. We need to remove all that. That is what the meaning of. We can do annotation. VCF tool annotates with the DB snips VCF with an ID annotated VCF filtered GVCF. A is called annotated file. C is called IDs. We need to give the variant IDs in the column. And then VCF file, we need to convert it into the tabular format. We need to give VCF file query -f, and we need to get chromosome position, reference alteration, quality, and depth. So this are the titles you just what. So you need to give that, and then you need to convert that filter GVCF into a variant format. Free bases. So free bases is a haplotype based variant calling method similar to GATK. So everything is command line based. So once we have download, we need to do the same process. So the same command lines. If you want to do a parallelizing, how you need to do a parallelizing stuff, how we need to run the variant calling. So here you can, if you want to call the variants, freebase -f faster, -m30, Q20, F is 0.2. What this Android mentioning? FNM is called minimum marking quality. The quality which is less than 30, we need to remove that. Less than minimum allele frequency. Allele frequency is less than 20%. 0.2 is called less. We can do the listing also. And if you want to do filtering with VCF file, here is called, sorry, VCF tools. This is called VCF tools. VCF minimum quality, minimum depth, recode, output filters. Minimum quality is 20. It's filter the variance with the quality score below 20. Minimum depth is 10. Filter the variant with depth below 10. Recode. It is helpful to provide the output file in new VCF file. This is a comparison with the BCF tiles and BCF tools and how it works. Join and based on the use, because once who get an hands-on experience to work with the GATK, they very much comfortable when to work into it because they can learn in and out of GATK pipeline. So if you want to learn that, go through the website and see in step by step of analysis and understand what is happening, what is the algorithm behind that, how it's working, what the each command line telling about, what is the process behind the development and delivery, what is it giving us output, how that output is looking like, everything you can learn. So make a look on that and then steady in the process. That is all I'll suggested. So what does mean by multiple sample calling is if we are having a larger net of data to call the variants, we need to analyze either based upon joint samples together by improving the main importance of during us main thing is we can able to see the variance which is shared across different samples, and second main thing is it is helpful to improve the genotyping quality. Third is it is helpful for better coverage across variants. The key steps to do the same thing is we need to align and need to call pre-process and we need to do joint genotyping and we need to do post analysis like filtration everything, and then we need to call the haplotype similarly for all the sample, and we need to merge the VCF what we have got it. So GATK combined GVCF reference faster sample 1 sample 2 we need to combine it to single, and then we need to perform joint genotyping generated unified VCF containing genotyping across all the sample. To do that we do joint genotyping, the output from the previous step, and we'll apply filter for that set of data. I want to do able to base BCF tools how this is. Oh, I didn't highlighted this. How this works, how the programs works. So, oh sorry. Right. I just give it a command and another column. So this is the command to run multiple sampling using different tools. In a quick Ofari I have added here. If you are working with deep variant, so coming into deep variant, variance is all about AI based, AI machine learning based, where it is providing high accurate variant calling when compared to GATK. So this is also quite good. So it is newly developed one. So it is helpful to do run the variant for a single results. Here is a command like what type of I already told. Then this is the comparison how accurate the data are in each approach based or speed based or scalability based or accuracy based or based on which approach it is works more efficiently when compared to other studies. Then how to manage VCF file format. So to tell about VCF files, so this is the output of called variant, how this is look like. So the VCF file having the header sections where it have the meta details about file content, reference genome, version, sample name and filtration. So you can able to see version, reference genome, sample content, everything. In a column header, it can define the structure of different data sections where it is telling like what is the chromosome, what is position, what is reference alteration, what if it was informations of the samples in the sample list are given. So you can see it then go into the data section. It can give this is chromosome 1 at position 1, 2, 3, 4, 6. G is G as in the reference genome is getting changed into A. Narration the quality is 50. This is good, and the filter is getting passed, and the information of changing the format, how how the allele frequencies, everything is getting mentioned. And you can see an operation when we are working with the VCF file. So to do filtration, VCF file filter I quality less greater than 30, filter VCF input file, exclude the futures. The excluded futures is like low quality futures and it getting shallow. And the specific to selecting the specific variants, it's a you need to if you want to view specific set of variants at from a position from the start position to the end position, we can able to do that through using the BCF tools view command. So where BCF tools view hyphen R chromosome 1, sorry, the chromosome 1 from a position this to this, and then in the input base here we can able to extract that data alone in the output VCF file. So where we can done through BCF commands and subsequent variants we can able to see. To merge the schools, we can the first main thing using worldwide is snippet. Snippet is also Java based tool where we can work with Java, XMLX, chart, snippet, chart. We need to give it like this, and then we need to give the reference, and we need to give the output input VCF file. As the end, you'll get it output like a annotated VCF file. I will give you a flow of how to work with different data sets. So I already built. So I'll share the flow. You can get it from each set of what is called each set of sliders I have shared. In that you have got that. So then you will work from the step-by-step process. Then what is annotation? Variant annotation is generally to do functional analysis, clinical interpretation, population frequency, or regulatory or structural components, or a pathway, are the gene interactions. This is all we are going to study as now. And sources over study, RefSeq or Ensemble, ClinVar or ClinVar with 1000 genome exacts or GenMat, or functional database that can be done with Snippet, CAD. If you want to do a regulatory database, that can be done with ENCODE. This all everything is a publicly open available database based on study, based on what we want to do. If you want to do a population based study, analysis, that can be mapped the details with the population database called Genomat, or XAS, or 1000 genomes so far. So another variation annotation can be done with ANOVA also. That is mainly for functional annotation. That can be done with the gene-based annotation, region-based annotation, and filter-based annotation. To do that, here is a command like annotate underscore variations. If you see, if you want to do ANOVA over there, before it is by type underscore ANOVA, but here is ANOVA annotate underscore variation dot Perl. It is Perl based. We need to give the output like annotated file. If you are working with human genome sample, we are giving HG 38, that is called latest reference genome, and input we need to give it as an input VCFL what we are working with. annotation tool is called VEP. VEP is called Variant Effector Predictor. It is developed by Ensemble tool. Yesterday, file like and we need to do a functional prediction and then are the main popular stuffs just to in calling variant annotation or variant annotation everything. So here I'm stopping. So if you have any questions to know what has happened, you can ask me. I know I went very fast to hide. Oh my god. So if you have any questions, you can ask. I'll ready to answer. It will be quite hard of variant calling, but when you do hands-on with the data set of what you have, that will be nice. Any questions? Is everyone is there, or I went very fast? What happened to everyone? I thought not to cover today, but I was very quick because tomorrow I thought to take machine learning based RNA sequencing analysis. What are the different machine learning techniques? How we can use machine learning techniques into the analysis? So that will be just add on to you to the course. So I thought I'll take into that. So meanwhile, if you have any questions, you can answer. Sorry, I can answer. Any kind of suggestions also, I'm ready to listen. So let me know anything openly to me. Any questions? No questions. All are very clear. No response. None. No one. No questions at all. Guys, are you there? Or you can't able to hear me? Okay, that's good. Thank you. Liana. Liana. Okay, Liana. Okay, if there is no question, we wind up today's session. We'll meet tomorrow, same 6 to 8 p.m. In that, we are going to see about how the machine learning techniques we can work with different types of data. So, what are the different types of machine learning techniques we can use in next generation sequencing data analysis stuff? So, that will add on to your course. Um, I don't go much into that. So, we'll stop and we'll see tomorrow. Thank you, everyone. See you. Have a good day. Take rest. Thank you. Thank you. It's okay. No problem.